Chapter 5: Chemicals and Cancer. This chapter examines how certain chemicals can initiate, promote, or advance cancer development. These chemicals may be found in our environment, workplaces, homes, or even our food. Some act by directly damaging DNA, while others interfere with normal cell signaling. Let's begin by understanding the different mechanisms through which chemical carcinogens operate. Chemical carcinogens are substances that cause or promote the formation of cancer. They are often categorized as genotoxic, meaning they directly damage DNA, or non-genotoxic, which act through indirect mechanisms. Some chemicals, like methyl-nitroso-urea, serve as initiators by mutating DNA. Others, like phenobarbital, act as promoters by encouraging the growth of mutated cells. Understanding these roles helps us evaluate chemical risk and develop prevention strategies. Cancer caused by chemicals typically progresses through 3 stages. The first stage, initiation, involves permanent changes to the DNA caused by exposure to carcinogens. The second stage, promotion, involves the clonal expansion of these initiated cells. In the third stage, progression, the cells accumulate additional mutations and become aggressive, leading to tumor growth and metastasis. Each phase presents opportunities for intervention and therapy. In the initiation phase, carcinogens chemically interact with DNA, causing mutations. These mutations can lead to altered base pairing, such as a guanine-cytosine pair being replaced by a thymine-adenine pair. If the DNA damage is not repaired, the mutation becomes a permanent part of the genome. This sets the stage for uncontrolled cell growth. An example is methyl-nitroso-urea, which causes GC to TOS substitutions. Promotion does not involve direct DNA damage, but instead stimulates the proliferation of mutated cells. Promoters like TPA mimic natural molecules such as DAG, which activate protein kinase C. This activation pushes the cell toward repeated division. Hormones like estrogen and testosterone can also act as internal promoters in hormone-sensitive tissues. Promotion is reversible, making it a target for preventive therapies. In the progression phase, cancer cells become more invasive and difficult to treat. Multiple mutations and epigenetic changes enhance their survival, motility, and resistance. The cells begin to dominate the tumor mass, outgrowing their neighbors. This stage is typically where tumors gain the ability to metastasize. It's also when cancer becomes clinically aggressive and much harder to manage. Chemical carcinogens are classified into several categories based on their structure and function. These include polycyclic aromatic hydrocarbons, PAHs, aromatic amines, N-nitroso compounds, alkylating agents, and natural products. Each class contains well-known carcinogens linked to specific cancers. Understanding these categories helps in regulation and public health awareness. It also guides research into safer chemical alternatives. Chemical carcinogens may be either organic or inorganic. Organic examples include benzopyrene, aflatoxins, and vinyl chloride, which are often found in industrial settings or contaminated food. Inorganic carcinogens, such as asbestos, cadmium, and nickel, are typically metals or minerals. These compounds act through different pathways and exposure routes. Both types are associated with serious long-term health risks. Many chemical carcinogens are not harmful in their original form. Instead, they must be activated by metabolic enzymes, most notably cytochrome P450 in the liver. This enzyme converts precarcinogens into reactive compounds that can bind DNA. Smokers tend to have elevated levels of these enzymes, increasing their risk. This highlights the complex relationship between exposure, metabolism, and cancer development. Once activated, carcinogens become electrophilic. They seek out nucleophilic or electron-rich molecules like DNA. This interaction can create DNA adducts, which interfere with normal replication and transcription. If these adducts are not repaired, mutations will occur. DNA is the most vulnerable and frequently targeted molecule in this process. The accumulation of these mutations eventually leads to cancer. Carcinogens are only found in labs. They exist in the air we breathe, the water we drink, and the food we eat. The International Agency for Research on Cancer, or IARC, has identified over 1,000 potential or known carcinogens. According to the World Health Organization, up to 20% of all cancers are linked to chemical exposures. These include industrial chemicals, pesticides, and pollutants. Everyday awareness and protective regulations are essential. Certain jobs expose workers to higher levels of chemical carcinogens. For example, exposure to aniline dyes has been linked to bladder cancer, while asbestos exposure causes mesothelioma. These risks have led to regulations enforced by agencies like OSHA. Personal protective equipment and proper training are key preventive tools. Occupational safety plays a critical role in cancer prevention. Chemicals like dioxins and DDT have been associated with breast and other cancers. These persistent organic pollutants are often stored in body fat and accumulate over time. Indoor air pollution from cleaning products and paints can also pose risks. In some cases, indoor air quality may be worse than outdoor pollution. Awareness and proper ventilation can mitigate these risks. Some prescription drugs have been linked to cancer. Diethylstilbestrol, or DES, once used to prevent miscarriage, was later found to cause vaginal cancer in offspring. Immunosuppressants like cyclosporin raise cancer risk by weakening immune defenses. Interestingly, some drugs like rapamycin may have tumor-inhibiting properties. These examples show the fine line between therapeutic and harmful chemical exposures. Animal testing plays a major role in identifying carcinogens, but species differences can complicate interpretation. For instance, saccharin caused bladder cancer in rats, but has not been shown to do the same in humans. These discrepancies highlight the importance of context and dosage in evaluating chemical safety. Human trials are rare due to ethical concerns, so researchers must extrapolate carefully. Scientific caution is essential when translating lab results to public policy.